Last but not least, it's my pleasure to introduce Dr. Lynne Ghorayeb, one of my own. She's a brilliant scientist in my lab, and she's going to talk about our efforts developing dual CAR targeting strategies for pediatric brain tumors. I specifically want to end with the title of her talk, From Design to Defeat, from really starting to clone a CAR all the way with the hope that we all together and the amazing science that we all showed today can actually defeat pediatric cancers. Lynne.
Thank you, Dalia. First, I'd like to thank the CCF committee for giving me the opportunity to present today, and I want to thank everyone who stayed late to the last presentation. I promise it won't take too long. First, I'd like to start with this image before discussing science. So let's imagine this patient being Parsley or Emma or any other children with pediatric brain tumors. These children are full of dreams, innocence, and energy. However, they are asked to face battles far too early in life. So throughout this presentation, I want us to keep Emma in mind and any other pediatric patients with solid brain tumors, because behind every data set, every experiment, and every advancement in immunotherapy, we are trying to come up with better treatment options for these children. So thank you all, and today I'm really excited to share our work on optimizing dual target B7-H3 GPC2 CAR T cells for pediatric brain tumors. So before I discuss our CAR T strategy, I'd like to first highlight the clinical needs behind our work. So as you may know, every day in the United States, 14 children get diagnosed with CNS tumors, and one of these children get diagnosed with medulloblastoma. So let's imagine Emma being one of these patients. For Emma, the standard of care would be receiving surgery, followed by chemotherapy and radiation. However, 40% of these patients relapse, and with relapse, we come up with very devastating side effects and very limited treatment options. So that's what drives the major need for novel, innovative therapies for pediatric solid brain tumors. So given the urgent need for more effective therapies, CAR T-cell therapy has emerged as a promising approach, and I'm sure you've heard a lot about CAR T-cell in this presentation. So briefly, CAR T-cell is a form of personalized immunotherapy that uses a patient's own immune cells to treat their cancer. So first, T-cells are isolated from the patient, and then they are engineered in the lab to express a CAR, or a chimeric antigen receptor. These cells are then expanded back in the lab and reinfused back into the patient, and then these cells are required to target a specific antigen on a tumor cells and eventually kill or lead to apoptosis of that tumor cell. So the CAR structure consists of an extracellular binding domain and other intracellular domains for activation. I'm sure also you've heard about the promising results of CAR T-cell therapy, specifically in hematologic malignancies. And one landmark example is Emily Whitehead, which was one of the first pediatric patients to receive a CD19 CAR T-cell and to achieve complete remission after she had failed her therapy with her chemotherapy for her beta acute lymphoblastic leukemia. So this suggests and shows that CAR T-cell is a promising approach. However, unlike Emily, many patients with pediatric solid brain tumors still face very limited treatment options. And this is because CAR T-cell therapy have yet proven its benefit in CNS tumors, and specifically in targeting or accessing the blood-brain barrier. And this is due to several challenges that CAR T-cells face, being tumor antigen heterogeneity, CAR T-cell trafficking and infiltration, the immunosuppressive tumor microenvironment, and CAR T-cell exhaustion. So we asked, how can we optimize our CAR T-cell therapy in order to come up with a better treatment options? So to address this, we came up with one promising approach, which is a dual CAR targeting. So first, why did we choose this approach, and what's dual targeting? I'm sure along the posters, you've heard about the CD19, CD20, and CD19, CD22 CAR combinations, which showed promising remission rates in hematologic malignancies. However, to date, there are no dual targeting trials for solid tumors. So with that, we aim to bring this concept into pediatric brain tumors. So let's discuss what's dual targeted therapy. So let's look at this single target CAR T cell. So first, the single target CAR can recognize a specific antigen on the tumor cell. Initially, it would effectively eliminate the targeted cells. However, there would be outgrowth of antigen-negative escape variants due to selective pressure, and this would eventually lead to disease relapse. So the loss or downregulation of the targeted antigen, being B7-H3 or GPC2 in our case, would allow the tumor cells to evade and lead to relapse. However, with dual-targeted therapy, and mainly B7-H3 and GPC2, we are aiming at recognizing dual antigens, and this would lead to effective killing of the tumor cells. There will be no outgrowth of antigen-negative escape variants, hence a more sustained and a more durable tumor control. So the concurrent targeting of B7-H3 and GPC2 would prevent antigen escape and promote a more durable tumor control and prevent the risk of disease relapse. Then the question is, why did we choose B7-H3 and GPC2 to be our main target antigens? So first, these two antigens are highly expressed across medulloblastoma cell lines, with B7-H3 that has shown to contribute to tumor evasion and resistance, and it has demonstrated clinical feasibility and manageable toxicity in many clinical trials. And GPC2, it's associated with potent cytotoxicity and durable tumor control in preclinical models. In these two graphs, they show the GPC2 and B7-H3 expression in red in group 3 and group 4 medulloblastoma and blue in healthy tissues. And as we can see, the high expression of both antigens in medulloblastoma tumor cells and a low expression on healthy tissues, which make them remarkably good complementary targets for our dual CAR approach. Now that we've chosen our strategy and our targets, the next step is to come up with the best CAR design. So we designed a panel of 9 different dual CAR constructs that differ in their hinge, transmembrane, and co-stimulatory domain, mainly for 1BB and CD28. And since these domains differ in their activation strength and persistence, we wanted to see which of these different CARs would achieve the best balance between activation and persistence, and hopefully move with this construct forward into the clinic. After we've designing these 9 different CARs, we wanted to make sure that our product is manufactured successfully. And in this slide, we can see a robust CAR expression of both antigens, B7-H3 and GPC2. We've assessed the CAR expression across the 9 dual constructs, but for simplicity, I'm showing a representative subset in this slide. So we can see a robust CAR expression of our CAR products, which means that our products were successfully manufactured and express both target binding domains. Now that we've assessed our CAR manufacturing, we wanted to see if they maintain a favorable T-cell phenotype. And in this slide, we can look at the heat maps showing the memory and exhaustion markers. Darker color mean a higher T-cell subset. And we can see that across CD4 and CD8 populations, our dual CARs maintain a balanced memory phenotype of central memory, naive, and effector memory, and even they maintain low exhaustion markers, which are LAG3, CTLA4, TIM3, and PD1, which means that our CAR products have a balanced phenotype and low exhaustion markers, which is favorable for a long persistence and a long anti-tumor control. Now that we've assessed our product and the manufacturing of our product, we wanted to see if our CAR designs could effectively kill the tumor cells. So this is when we undergo a cytotoxicity assay in which we plate our tumor cells and then we challenge them with our dual CARs. We incubate them for 48 to 72 hours and then read it with an NTS reagent. And the graph to the left that shows the percentage of live tumor cell to the effector to target ratio, the effector being the CAR T cells to the target being the tumor cells, we can see that our lead dual CAR represented in red maintained its cytotoxicity at high effector to target ratio and even at lower effector to target ratio, meaning that our lead CAR design is effective in killing and has a good cytotoxicity profile. Now, the figure B, which shows the fold expansion to the stimulation number, this is done with a persistence assay in which we plate our tumor cells and then we challenge them with the CAR T cells and read them after 48 to 72 hours, and we keep replating our CAR T cells until they lose expansion or killing, and we can clearly see in red that our dual CARs maintained a strong persistence up to 5 stimulation as compared to the single CARs and the control CARs. Now that we've seen that our CAR products are successfully manufactured, they have a good cytotoxic profile, and they maintain a balanced memory phenotype, we wanted to evaluate this in pediatric orthotopic models. And this is where we established an orthotopic medulloblastoma tumor model in which first we inject our mice with tumor cells, and then we treat them depending on the tumor model using, and then we assess tumor burden and survival. In this in vivo experiments using group 3 medulloblastoma cell line, we can see that some of our dual CARs, mainly the one that I circled, maintain a favorable anti-tumor control, and this is shown by a reduction in the flux. After we've seen this good potential with this first in vivo experiment, we expanded this to group 3 and group 4 medulloblastoma, mainly CHLA, MB002, and D556 tumor cell lines. And in this other experiment, we can also see a sustained tumor control with our dual CAR and an enhanced survival shown in red. And this is another experiment also showing a sustained tumor control and enhanced survival. And currently, we are still ongoing many in vivo experiments, and not only targeting medulloblastoma, but also focusing on other tumor cell lines. Now that we've seen that our product works in vivo and in vitro, the next step is to hopefully translate this into the clinic throughout the DEFEATED trial. So the DEFEATED trial is led by Dr. Roger Packer as a principal investigator, with Dr. Dalia Haidar as a scientific lead investigator, bridging between the preclinical and the clinical development, and the IND is sponsored by Dr. Catherine Bollard at Children's National Hospital. So we are really excited to open a phase 1 dose escalation study in which we will be treating patients with relapsed or refractory group 3 or group 4 medulloblastoma cell line. And this would be a dose escalation study mainly focusing on the safety and feasibility, since we are injecting or treating the patients for the first time with this dual B7-H3 GPC2 CAR T cells. And also we're assessing other exploratory objectives such as the antigen expression, the anti-tumor activity, and overall survival. Now with that, with our dual targeted therapy, we are aiming at reducing antigen escape by lowering the chance of tumor cells evading the treatment, enhancing therapeutic efficacy by targeting the tumor cells via 2 distinct mechanisms, by increasing tumor specificity by targeting 2 antigens and reducing the damage on normal tissue, and then having synergistic interactions by combining, which would enhance cytotoxicity. And with that, I'm really excited to mention that we are submitting our IND by the end of this month and hopefully opening our phase 1 clinical trial by the end of the year. And hopefully for patients like Parsley, Emma, and any other children with pediatric brain tumors, we will be able to come up with better treatment options, and the patients will no longer be on different battlefields, but on the same path of healing. And with that, I would like to thank specifically Dr. Dalia Haidar for her incredible mentorship and vision that make this work possible, the Haidar Lab for all the support, and specifically the CIR team and the SETI team, and everyone working on this clinical trial to make it possible, and all the funding. Thank you so much.
Thank you so much, Lynn. Any questions to Lynn? Yael?
So exciting and really beautiful talk, and data are very compelling. Are you going to be delivering the CAR T cells directly into the CNS? And
Yes.
Are you going to be giving lymphodepleting chemotherapy?
Yes. Yeah.
Excellent. And Dalia, appreciate you.
Sorry, and I have to. Yeah, we are only giving 1 cycle before the first infusion, but not for the successive infusion.
So that's what my next question is. You'll be doing repeated infusions.
Yes. Lymphodepletion.
Excellent.
Any other questions for Lynn?
Really cool work. I was curious, I don't know if you specifically didn't say because you don't want to say, but which of your co-stims for your 2 different targets ended up being best?
Yeah, so our best dual CAR till now has the 28Z costim domain, but we're still undergoing many in vivo experiments just to validate that. But yeah.
Ask not to share. So, but yeah.
GPC2 binder. Which binder are you using?
Yeah, the SCIV, the GPC2. Yeah.
There are several out, and so some
yeah,
are.
This is our own binder.
Yeah, it's our.
And do you know the epitope on GPC2 that it's recognizing? Do a comparison of the GPC2 binders.
Actually, yeah, we have different constructs, and mainly it's the DY19 SDIV for the GPC2. Yeah, it's almost.
Can you talk about your next study about?
Yes, for this one, we only use one specific binding for all the car designs, but we are aiming at using different epitopes for the CHOP study. Like we are collaborating with another team that we will be using another epitope. Dalia, you can comment on that if you.
Nice variant. That's and others that don't. So.
I'm sorry, I do have to jump in because I designed the cars before. Yes, it does bind to the same epitope like the GPC2 binders that are in the market. We did not do like head-to-head comparisons with the dual cars in terms of the GPC2 binders, but that is planned. We are working with John Mattis's group and Jessica Foster at CHOP. They have other dual cars, and we're going to be doing head-to-head comparisons. Yeah. But we did compare it at a single car level, the GPC2 versus the GPC2, and they had very similar performance.
With the two targets, do you anticipate twice the CRS or increased CRS, or what do you anticipate?
Yes, so we anticipate an increase in CNS or cytokine release syndrome since we're targeting both antigens. However, this would be a major part of our clinical trial, assessing the safety of our product, and currently now we're doing toxicity studies just to assess the safety. But also targeting both antigens, for me, it will increase specificity and maybe decrease of target response, and this would maybe help in the safety profile. So.
Dual cars, now you have lots of co-stims, like twice as many co-stims. So what's the story with exhaustion?
Yeah, so mainly.
I mean,
I'm sorry, can you just repeat the question?
If the co-stims are involved in exhaustion of the CAR T, then my naive understanding of this whole field would say that if you have twice as many co-stims, which you have in your vector, would this really amplify your exhaustion?
I believe.
Or propensity to exhaust?
Yeah, and I believe that's why we are designing different dual CARs, and our lead CAR construct is the one having we are aiming to balance between using the 4-1BB and the CD28 co-stim domain to have the best balance between activation and persistence, because exhaustion is one of the main drawback for our CAR T cell.
Any other questions for Lynne? Can I ask you one? How many mice did you implant for this? I'm kidding. Okay. Well, thank you so much, everybody. And with that, I would invite Bridget to close up.